Thank you for having me. I'm really excited to be able to present to you today, but also listen to the very interesting talks that we have on the agenda today. But first, as you know, the RCPA QAP team has just flown in from Sydney, Australia. And at the moment in Sydney, the Vivid Festival is on, a festival of light and music and art. And we're quite impressed by our Vivid Festival. But when I came in on the weekend, I realised that so many of your impressive buildings are lit up all the time, and it looks like this city is really one that is very festive, and we've had such a great trip so far. But we can't talk about beyond compliance without first having a look at ISO 15189 and the requirements of that standard. I'd like to address the differences between EQA and QC and just how we can use EQA to mitigate the risks to patients. And really that requires understanding the limitations of your assays and being able to identify erroneous results. Is there evidence that EQA participation improves patient safety? For this, we'll have a look at the literature and then just a quick thought on EQA's broader role in patient safety. So what are the requirements to be compliant with ISO 15189? Well, it starts at the top with the organisational structure and ensuring that it is well organised and roles and responsibilities are clearly defined and that there's adequate supervision of laboratories by a qualified pathologist. There's a robust quality management system that has documented policies and procedures and records of corrective actions to reduce incidences. And that the external services that the laboratory uses to refer tests to are also compliant to ISO 15189, and that there's a clear process for selecting suppliers and ensuring that they're compliant as well. In the laboratory, you really are an advisory service, so the framework covers how you would communicate with your customers and how you can, they can provide feedback to you and how you handle that. And then, of course, identifying and controlling non-conformances and a record of how your corrective actions are captured, how a thorough root cause analysis is conducted, and what kind of preventive actions you can prevent in your lab to control non-conformances. And of course, audits, our favourite. So lots of internal audits happening, but also those external audits that can allow for continuing accreditation. And your technical requirements, that you have competent and highly trained personnel that know their responsibilities and role in the laboratory. The laboratory equipment that you select has been thoroughly validated and is in an environment that's going to help it perform to its best. There's the pre-examination and post-examination processes, ensuring that those samples are collected correctly, transported and stored appropriately before testing, and that the correct methods are selected for those tests. And of course, QC and EQA participation, which feels like it never ends sometimes. So I'll talk about that in more detail today. And post-examination, ensuring that those results are reported accurately to clinicians and safely stored in your laboratory information management system, securely, confidentially, and with traceability. So that's ISO 15189, but I'd like to talk about the differences between QC and EQA, and I'm sure you're all very familiar with this. We know that QC is used every day on your run to ensure that you're getting the same result on your run as you got yesterday, and if your QC are out of limits, what do? You stop the run, don't report those results, and you go and investigate and make sure that your run is back in control before you're releasing results. But EQA really is about quality improvement. It's a way to harmonise and standardise the results that we release. It provides post-market surveillance of our methods to make sure that they're still accurate and precise. And so it's not just about analytical quality. And this table here shows a quick summary about the difference between EQA and internal QC. So, like I said, internal QC can make sure your results are precise and that they're same as they were yesterday, but really, is it the right result that you're putting out? Only EQA can truly tell you that. Both can control bias in your system as long as your internal QC limits are set correctly, but EQA really can provide information on the linearity of your assays. Your internal QCs hopefully have a QC at the bottom end of your assay and the top end, and hopefully somewhere around your clinical decision point. But a good EQA program should really cover the entire linear range of your assay. EQA is there to provide robustness and to show you where the risks are in your assay. But QC, internal QC, provides that real-time alert, and that is what's so valuable about QC. And more we're seeing EQA programs that can provide a real-time assessment. And the Holy Grail is a commutable material, which is variable for both QC and EQA. So EQA really is an investment in reducing the risk to reporting incorrect results and minimising potential harm to patients. But like Tony said, it really is your role to work out how you can do things better in your lab. EQA is a significant cost to a laboratory, so you really want to make sure that you're getting the value out of that EQA program and not just looking to see if you're right or wrong. So let's look at the ways you can reduce risk to patients. And using EQA allows laboratories to go beyond compliance. When you're compliant to the standard, you've really met those minimum requirements. But EQA provides an external benchmark of performance against your peers, allows you to identify systemic issues that internal controls may miss, and it really fosters a culture in your organisation for wanting to continually improve and increase your training and competency, which ultimately enhances patient safety and care. So we see many issues in our RCPA, QAP, EQA programs, and I just want to touch on a few of them today. I wish I could talk about all of them in more detail, but the common issues that we see are due to a lack of standardization in assays, even assays where there is a reference material available, and we see lot-to-lot variation due to this lack of standardization. Lack of harmonization of testing algorithms. We all do it a little bit differently in our lab, and that affects interpretation of result. The reference intervals that a lab uses and the cutoff values that are established in that lab are a source of issue that we note in our programs. Then there's the missed interferences or even an antibody masking the antibody of interest in our assays, interferences like hemolysis and lipemia. And EQA can really help with method surveillance. So is my method the right one? Is it the best method that I could possibly use in my lab? And how is it performing over time? Another favourite of mine, being from immunology, is that issue of antigen excess or prozone phenomenon. It's been around for a really long time, but we still see it affecting results in the lab. And of course, missed rare or unexpected antibodies, microorganisms, or gene mutations that QC just won't be able to detect. So that really is where the value of EQA comes into effect. So here's a little video of our lab, just a snippet of what you saw earlier. This is our logistics manager, getting your transfusion samples ready, and our team, our hematology and transfusion team, boxing them all up and hoping to get them to you in a good state for testing. And that was the transfusion program where we've recently identified some rare or expected antibodies. They're difficult to detect because they're infrequent. So we do aim to provide these rare or unexpected survey challenges occasionally in the program just to help you identify and address those gaps that you might have in your detection capabilities. So this was survey 3 of the transfusion compatibility program this year, where we saw participants using panels that included Asia cells, and they were able to detect not only the target anti-S antibody, but some labs were also reporting the anti-MIA antibody. So you may have been familiar with this survey. So the incidence of the anti-MIA antibodies is rare amongst most populations. However, there's higher frequency of occurrence noted in the Chinese and Southeast Asian populations. And when we investigated the survey results, we noted that some customers in Southeast Asia were noticing this additional antibody that even we weren't expecting. So our Australian Red Cross Blood Service, where our blood comes from for the samples for this program, currently doesn't even actively screen for anti-MIA antibodies. And since most Australian labs don't have this antibody identification panel, this antibody could easily be missed in our community. And so to address that, thanks to our neighbours overseas discovering this antibody in this sample that we sent out, we've decided to order panels of cells that include the MIA cells. Again, I mentioned the antigen excess or prozone phenomena or hook effects. It occurs when the antigen is present in such high levels that it limits antigen-antibody binding, resulting in formation of these smaller immune complexes, causing immunoassays to underestimate high concentrations of protein. So how can you understand the limits of your assay to know whether this is a possible effect? Well, really, where there's a very large physiological measuring range, so values that can range from less than 1 milligram per litre to over 10,000 milligrams per litre, that sort of assay is really at risk of antigen excess. And we saw that last year in the rheumatoid factor program. So this is a local lab who we were expecting a value of 200 and returned a low value of 137. So they were marked as discordant. And what would we expect you to do in this case? We'll really go and investigate why your result was so low compared to your peers and all results. But this lab only reported a qualitative value, a positive or negative, and their result was still positive, so they didn't worry about investigating the difference in the quantitative value. Then when we were expecting them to return a value of greater than 700, it went even lower, 100. But again, it was still positive, so they didn't feel the need to investigate this too much further. And of course, by the end of the cycle, when we repeated those levels, they were still low. And you can see their linearity compared to the median is very, very low, and their linearity compared to all results, but also their subgroup, is very, very low. Which makes you think that it's not just the EQA sample, of course, but also it might not just be their method in this case, and due to method variation. So the lab contacted us, and we explained this to them, and they went back to the raw data from their analyzer and realized that, in fact, a Z flag was there on their raw data. It's just that the scientist that was authorizing these results wasn't sure what that meant. So they've since been able to correct that issue, and now they see the value of reporting not only the qualitative result, but also the quantitative as well. And really the issue here is that, sure, it was still positive in this instance, but a result of 2,000 in a patient could have equaled a 0 result. So there is a risk to patients there. We see lack of standardisation in assays coming across in so many of our programs at the QAP. I've got an example here of the sweat test chloride measurand. This is where we give a sample of varying levels accompanied by some clinical information that will help a lab decide which cutoff to use for this screening test for cystic fibrosis. So you can see in sample 6 this year, we expected a level of 40, but actually the results returned, the qualitative interpretation of the results were very varied. And this was in a 3-year-old female. So most labs did report this as equivocal, but many labs reported normal and some positive. And you can see the issue that would cause in a screening assay of a young child. So what are we going to do about it? So the RCPA QAP sweat test program has highlighted differences in the cutoff values established in laboratories that are participating in the program. We know it's a problem. We know that cystic fibrosis cases have been missed in Australasia due to this lack of adherence to guidelines. And so the AACB, in collaboration with the RCPA QAP Advisory Committee, has undergone a further review of the guidelines, and there may be a case to move the cutoff from the current 40 to less than 30 millimoles per litre for the guidelines, and this would be in line with the new CLSI guidelines for this as well. So it's a delicate balance. This may prompt more false positives that then require genetic testing, but I think the argument is that it's more favourable than missing cases. So this is evidence of how an EQA program will have a wide and positive impact on patients. And this is an example of real-time EQA. So this is our COVID-19 antigen app that you might be familiar with that's really useful for method surveillance. So in the early days of the COVID-19 pandemic, when there were a lot of new assays on the market to detect COVID-19 antigen, there really was a need to ensure that the results that were released were reliable. So we quickly developed an app where you could enter the name of your test, submit the results of the samples that we provided, and include a photo of your rapid antigen test, and get an immediate result. And I have to say that Malaysia was really the leaders in EQAing their COVID-19 antigen, and we're sure that this gave your health authority the confidence in the results that were coming out of the rat test and really helped to inform the management of the pandemic in those early stages. And then came the EQA program for the RNA detection of SARS-CoV-2. This EQA program was mainly aimed at Australian and New Zealand labs and gave confidence in those early in-house testing methods, and then the early commercial assays that came available were actually providing correct results that health authorities could act on, and the risk to patients in the community we know would have been so high if there wasn't an EQA program for these tests. Method surveillance has been an important topic for procalcitonin for a long time. Procalcitonin is a useful marker for antibiotics and assessment of sepsis risk. And we do see a lot of variation in this program, not only within the quantitative users, but also the qualitative users. So you can see in sample 4 last year, the quantitative target was 3.44 nanograms per mil, but we saw a lot of variation, which I'll show you on the next slide. And then the semi-quantitative procalcitonin users had quite the variation around their cutoff. So this is the 2 clusters of quantitative results, and that was for our Abbott and Roche methods that returned lower results, and our Beckman and bioMérieux and Siemens reagents that returned higher results. And this is such a critical marker, not only for the patient, who obviously you want to reduce the risk of developing sepsis by introducing antibiotic therapy, but also more of a global responsibility around correct antibiotic stewardship. So it's really important to get this one right. And we can see here in sample 4 last year, but this is also not uncommon in so many of the surveys that we send out, that there was a split in the semi-quantitative users around a very low risk of sepsis and a high risk of sepsis. So I think in your investigations, you would be wondering whether this method was fit for purpose. And there's a few papers on the harmonisation of procalcitonin, with Tony, I think, as a co-author on this one, and our team has recently submitted another paper on procalcitonin. So interferences are probably well known to you in your lab. We have the serum indices program that monitors the effect of interferences in our samples, so covering hemolysis, lipemia, and icterus samples. And what we've seen in this program is that manufacturers and participants really don't have a good understanding of how serum indices should be used. We see so much variation in the units and indexes, and not really sure how to troubleshoot these samples. And not only do we see inconsistencies in the program, but we see inconsistencies even within the own hospital network. And this is such a risk to patients because many laboratories base their reject or accept criteria solely on the manufacturer's recommendations. There's the potential for inappropriate reject or accept decisions due to interferences, and you can imagine how that impacts patient care. And there's the potential for unnecessary recollections, which is not only invasive to your patients, but it does have a financial burden for your health system and also results in delayed treatment and monitoring. And there's the potential for inappropriate treatment when some compromised results are released, and we see this in elevated LD due to mild hemolysis. So in our liquid serum chemistry program, we had a look at some of the report comments that were coming back for potassium and LD. And you can see here that in a sample, it was sample 4 in 2022, that had a hemolysis index of 53 milligrams per deciliter. 5 labs reported the potassium with no comment at all, which probably wasn't appropriate, and we know that an incorrect potassium result can be quite critical for a patient. And 10 labs reported the LD result with no comment at all, which if a hematologist saw that and thought maybe there was a relapse of leukemia, it could be a very serious erroneous result. And still on interferences, the CSF program for oligoclonal bands provides us with a lot of discussion around interferences. And this is an example of a sample from 2017, but actually we see this time and time again in the program where there is variation in the reporting of oligoclonal bands. In this case, we expected an absent report, but many labs also identified oligoclonal bands. So we got some participant feedback who said that they were using the isoelectric focusing method, but because the hemoglobin in this sample was so high, it seemed that it was blocking the anti-IgG staining of polyclonal immunoglobulin, creating an artifactual oligoclonal picture. And they weren't quite happy that we included this challenge in the program. They felt that if this was a patient sample, they certainly would have seen the degree of hemoglobin in the sample, but because it was loaded onto the gel from a brown bottle, they missed it, but did agree in the end that it was a learning moment for them. And so we included some additional commentary. The ACB CSF Working Party made the following comments: that this was a grossly hemolyzed sample, but in fact, no monoclonal IgG was added to simulate oligoclonal bands, and that this challenge was really included to allow participants to distinguish between oligoclonal bands and artifacts. And if you've had to run this test before, you know how precious the CSF sample is. This test is used to diagnose multiple sclerosis or Guillain-Barré syndrome, and to obtain the sample requires a lumbar puncture, which is extremely invasive to a patient and not one that a doctor's likely to recollect. So it's so important that the first result is the right result. Another example of where QC had missed an issue, but EQA was able to pick up an issue with a lab, was noted in the point-of-care blood gas and co-ox program. The participant was consistently showing a negative bias in their results. I think they assumed that it was due to the EQA sample because their QCs were still within acceptable limits. So they kept running and reporting patients, even with an APS of 2, Z-score of negative 5.4. So we suggested a patient correlation just to confirm that it wasn't the EQA sample, and sure enough, their patients were low as well. And you can see their bias, negative bias down here with their result, and probably a few dots around them were from the same method group as well. So they locked the instrument as soon as they completed that patient correlation, and radiometer went in to investigate and found there was in fact a faulty hemolyzer, which was replaced for this lab and those others affected, which are probably represented in these dots here, these outliers. And that was a very serious issue that would not have been picked up with QC. And I wish I could talk a bit more about lot-to-lot variation because it is such a huge issue for laboratories. It's difficult to manage, and we do see that cropping up in our programs all the time, and the need maybe to establish new internal cutoff values. When there is a lot change, we see shifts in the medians of quantitative results, which could have an impact on your decision limits, and we see in qualitative results that it could lead to false positives or false negatives. So a thorough evaluation of a new lot is always recommended before accepting it from the manufacturer, and just to reduce the variation, selecting a lot with a long expiry, and in some cases, a factor may even be introduced to standardise results following a lot change within a lab or even across a network. And sometimes a population-based cutoff value may need to be established rather than just relying on the manufacturer's reference ranges to ensure that results that you're reporting to your clinician don't change significantly with a lot change. So we've had a look at just a very brief touch on all the ways that we identify risk to patients with the test that they're running. And we see this in the EQA, the RCPA QAP EQA programs, but is there any evidence that EQA participation can actually improve patient safety? So I had a look at the literature. This paper from the Asian Journal of Transfusion Scientists is a lab that did a 5-year review on their participation in the laboratory and did find that it improved their participation. They also were able to identify an anti-MIA antibody that had previously been missed. Here is another EQA provided for COVID-19 testing where EQA participation showed a marked improvement in the rat tests that were being provided. This was a Canadian study. And the Journal of Clinical Microbiology, where a paper was published on the quality of HIV viral load testing using per-efficiency test programs. And this was an EQA program initiated by the CDC, providing a dry tube specimen in a resource-limited setting that was sub-Saharan Africa, which was really useful for identifying HIV viral loads accurately, which could then inform antiretroviral therapy. So again, another example of where EQA is giving the confidence to not only manage the risk to the patients, but to the community and possibly the world. So a brief look at EQA's broader role in patient safety. We know that it can identify discrepancies and areas of improvement. We know that it can help a lab develop and adopt standardized diagnostic criteria. It can provide benchmarking data, so you're not just trying to meet the minimum standard, but you're really trying to become the best lab that you can be, and allows you to identify systemic errors like reagent issues and instrument calibration problems, or even the technical competency of your staff that QC often misses. And a good EQA program should really provide you with an educational component so you can get the best bang for your buck. It should provide feedback, it should be useful for training your staff, and really deploying best practice guidelines to your laboratory. This slide is included just for those of us who like to see a list completely checked off. And finally, I would just like to pose the question to you: how has the RCPA QAP EQA program uncovered an issue in your lab that you are unable to detect with QC or otherwise? I invite you to have a look at the paper in front of you. There's a QR code here that will take you to the Slider platform, where you can pose any questions that you have throughout the day, and they'll be addressed in our panel sessions. But if you would like to tell us how EQA has helped you in your laboratory, you can pose that in the Slider platform. And if you see a question that you would really like answered today, give it a thumbs up, and our panelists will know to address that question. Thank you.
Thank you, Zoe. So, as well as using the Slido to put your comments in, if there are any questions you have, you can use the Slido thing. That way, you know, it's much easier for us to get your questions. As Zoe said, we'll go through them later, and they'll be answered at the ISO 15189 session. So thank you, Zoe.